does go up and down a lot, naturally. However, if you look at the end, and on the next slide I have a closer view, it goes up significantly in the last 150 years or so. It's believed that is anthropogenic factors because of the Industrial Revolution and the introduction of burning fossil fuels. And that's as I said, so that's a closer look there, and the spike is huge compared to what was there before. And so, as I said, the impacts of fossil fuels are causing these large spikes in CO2 levels, as we suspected. And so that releases greenhouse gases such as carbon dioxide. Others include water vapor and methane. But what effect does this have on us and the Earth? And so the most research into these are global warming and ocean acidification. Now, global warming, I'm sure we all know, is quite simply the warming of the globe through the greenhouse gas effect. And so light comes in, and some of it is absorbed, and some of it escapes. That's a normal process. But with increased CO2 levels and other greenhouse gases, the heat is absorbed more, and so the temperature goes up further to an unnatural level. And ocean acidification, I'll go on to the next slide here. There's a good paper by Scott Doning and others. It's really about the fact that, yeah, sure, global warming is a big problem, but ocean acidification is fairly overlooked, and sort of the other CO2 problem. I do recommend a read. And it really looks into the effect of organisms, such as like mollusks, and all those there. And it's really quite a significant factor. And so my project was to look at how carbon dioxide affects certain water chemistry elements, such as the pH, phosphate, nitrate, and calcium. And so I'll describe to you how I did that. And so the methodology here. Now, it's very important to have a reliable and reproducible project, purely because other people can do it again themselves in order to compare to different sites and different times of the year. And so with mine, I made sure that all equipment was cleaned. Now, the Marine School, we all share our equipment, and so that can be good and it can be bad. It means that someone before you has used it, and so it needs to be cleaned properly. And so in order to do that, when I took the initial test samples, I kept a bit of water on the side, just so I could rinse everything down. So I wiped it down with sample water, and that makes sure that no contaminants get into it, or contaminants that aren't present in the sample water anyway. And each test, so the test that I took for pH, calcium, nitrates, and phosphate were all doing the same time. I was very tedious with it. It was all written down the same way. Even though they had instructions, I made sure I shook it the right amount of time each time, and it was really quite boring. And so the test took place between the 5th of February and the 24th of March this year. And so that is quite a large scale, but due to the fact that I only have one tank for all the tests, we can only do one test a day, as it's over 24 hours. And so each time I got the water from Prince Wales Pier. For those of you that know Falmouth, it's just down by Boots and near Tesco. Lovely pier, recommend it. And so I carried that about 15 litres back to the Marine School, and I then did the rest of the project here. And so I started with preliminary results. So they were taken in January just to see how everything works. And then I took the first test for once the water had been gathered, filled the tank, and also retained some for testing. And so they involved the pH, the calcium, the phosphates, and the nitrates. And so the tank roughly held about 40 litres, and so 50 litres went into there. And I marked simply on the container where the water that I was going to fill up each time, so it was the same amount, and the level on the tank, just to make sure it filled up to the top. And if it didn't, I'd go back and get some more, if necessary. And so with the actual setup, it was a slate rock. It's quite easy to clean. And so I had that at the bottom, and then on top of that was an egg crate board, which is just a plastic board with lots of holes. And on top of that was a beaker, which I put in, made sure all the air was taken out, then put upside down. And so the reason that it was on this egg crate board is so that the CO2 that's going to be put in the next stage is able to diffuse out the water there. And so and then there's a hose connected to this little CO2 cylinder. It goes under the lip of the beaker, turn the valve on slowly, so as to not break anything, and then to the indicated volume of carbon dioxide that was needed. So that was either 100 centimeter cubed, 200, or 300. And as I reached the end, obviously turn it off. And so, as I just said, the volume there. So then after that, I'd taken all the tests, I then sealed the container, left it for 24 hours to allow the carbon dioxide to diffuse into the water. And after those 24 hours, I then took the same test once more in order to compare from before. And each test, so say pH was done 4 times for before and, the experiment was done 4 times for each volume in order to point out any anomalies or outliers in the results. And so I'll take you through how I did nitrate here. So they're all fairly simple. They came in their kit with their own instructions. And I did this one first, as it was 5 minutes. It just was logical to do that first and then do the shorter ones after. I could do it quite efficiently. And so it's simple, using an API fish care kit, which you can buy quite happily. And you drop a few of the sample solutions in. It changes colour, you shake it, you leave it for the time. And at the end you have a card that just shows you the different colours, indicating different levels of nitrate in there. And so you leave it for 5 minutes and compare. So to do that I had the sheet itself to compare to, the solution in a clear container, and just a white bit of paper. Held it up to the light so you can see, really get a proper indication of what the colour was. And this, as I said, this test was taken before and after each experiment. And so then moving on to phosphate, which was also done before and after. This was shorter, 3 minutes, so once I've done that, move straight onto this. Quite simple because I can get it done as quickly as possible, but with still optimal results. Same place I got this test kit from, and again, same colour comparison. So it's all quite a similar methodology for each test. Moving on to pH. pH takes no time at all. Any of you have done a pH test before, it's just 7 drops of the indicator solution, mix it about, and again, compared to a colour comparison card. And so that could be done very quickly in that time. Calcium, however, was a bit more delicate, as it was a titration. And so I would do that after the 5 minutes had all finished, so I had enough time to do it properly. And so with this one, you add certain amounts of powder or drops, and the last stage is the titration. We have 1 milliliter of water, 1 milliliter, sorry. And at this stage, I added 0.6 of it in, making sure it's thoroughly stirred after about 0.2. And at this point, I then added 1 drop in each time, because the colour change happens fairly quickly. Although with this one, I must say, it did go from, well, it's supposed to go from violet to an indigo. And it did kind of have stages in the middle. And so I dropped until it was the dark indigo colour. And as, same as the others, taken before and after. These are the result tables. Won't stay on this slide long, as I've got them in my research paper, if you'd like to have a look at them. But the graphs show a much better observation of what really happened. And you'll see them again as I talk through it more. And so the calcium, it's clear that you can see here there is a definite change after CO2 has been introduced. I think you can all see that here at the bottom. The most significant by far is the 300 centimeter cubes. You can see it's dropped by about 60 parts per million. I think I have it here. Yeah, with a difference of 63.75 parts per million in over only 24 hours, which is significant. That's huge. And a lot of organisms depend on it. On calcium for different processes. One process that calcium does in the ocean is that it acts as a buffer. Now a buffer maintains pH within a certain band, whether that's alkaline or acidic. And so the natural pH of the ocean is between 8.2 and 8.4 on the pH scale. And the buffers help maintain that. But with less calcium after carbon dioxide was introduced, it means there's less calcium to buffer. And so we're going to explain pH later on the slides here. But calcium is also very important in mollusks, such as bivalves, who use calcium for their shells. And if there's less calcium, it means weaker shells, or less shells, which means they get predated on. And that simply has a knock-on on the ecosystem. And so if there's less of a food source, there's less of something to feed on, which means they'll have to die off or find another food source. And as that moves on, that could have a knock-on effect on higher trophic levels, and eventually even us. And the pH. Now, pH here, you can see, is a huge change in just the 24 hours. I mean, even 100 centimeter cubed, you drop about half a grade of the pH scale, which would cause phenomenal effects to us. The most significant is, of course, at 300 centimeters cubed, where it drops by an entire unit on the pH scale. It goes from alkaline to weakly acidic, which would wreak absolute havoc to the oceans. Now, it dropped over 24 hours, and of course it's important to remember in mind that the carbon dioxide that I've introduced to this project is significantly exaggerated to what we see in the real world, but it's merely to show the effects of carbon dioxide on the water chemistry. And so that would not happen so quickly in the oceans at all. It would take thousands of years, but that's not saying we should ignore it. And so, as we were saying, the pH affects many things, lots of biological process, and if anybody knows anything about proteins or enzymes, you know that pH can change the structure. And so the hydrogen ions just destroy the structure. So it's still there, but it's different. And if it's not the same structure, it does not work at all. And so if that was to change as it did at 300 centimeter cubed, then nothing would work. Everything would really just die out, apart from some small bacteria that may be able to survive due to their conditions anyway. And so the phosphate also dropped a fair amount. You can see again, obviously the most significant is at 300 centimeter cubed, with a total difference of 0.225 milligrams per litre. Now considering it started at about 0.3, 0.35, that's huge. Like I calculated, that's about 78% change in 24 hours for phosphate present in the water. But what does this mean to us? Well, CO2 is very important for plant growth, and that equates to algae. And less algae has, well, if algae can't grow quite so much, it means that there's less primary production, which is simply photosynthesis, which puts oxygen and provides energy for the marine organisms. And with less primary production, it means energy is not passed on through the whole food chain. And so, if the primary consumers can't get energy, it means secondary and tertiary can't get anything. And if that means nothing can get energy in the ocean, it means we don't have food as well, which causes work. Well, especially with growing populations, if we don't have enough food, that's going to be a big problem. Now the nitrate's a little more interesting because it showed nothing almost entirely throughout the experiments, apart from one result there at 370 cubed, which has skewed the average very high. It was 2 milligrams per litre, which was unusual because I cleaned the equipment each time, and it was just the one day, the one test. And I tested it a couple of times, and I got 2 milligrams per litre, which skewed it up. Now, I think the reasoning for that must be because of contamination in the water itself. It's possible that it was from human error, but I believe myself, it's more likely that it's from the water that day. Maybe there's some runoff. So there were the 4 observations that I intended to make. Now, with algae, as I mentioned before, I've just gone over a little bit. Carbon dioxide helps algae grow. That's well known. But during this experiment, as I introduced more carbon dioxide, the algae growth was phenomenal. 100 centimeter cubed, it was a bit of a bother to get off the kit. Not a problem, get it off. 200, a bit more of a problem. 300 centimeters cubed, the entire tank was caked in algae, just up to the water level. And so that required more hardcore cleaning products, so to speak. That's just saying that. So what can I draw from this? Well, it's clear that the carbon dioxide certainly affects the pH, the phosphate, and the calcium present. However, the nitrates, it's inconclusive at this moment in time. I think I would like to have a look with more precise equipment that looks in decimals, so 0.001. But as of now, I have to draw the conclusion that carbon dioxide does not affect nitrates. But it also draws the fact that it has a significant effect on photosynthetic life present. And that's what I have to say for you today. Have you got any questions?